Smooth Chapter 1, Section 10, Smooth Muscle. Smooth muscle lacks striations, which distinguishes it from skeletal and cardiac muscle. The striations found in skeletal and cardiac muscle are created by the banding patterns of thick and thin filaments found in the sarcomeres. In smooth muscle, in contrast, there are no striations because the thick and thin filaments, while present, are not organized into sarcomeres. Smooth muscle is found in the walls of hollow organs, such as the GI tract, the bladder, the uterus, as well as in the vasculature, the ureters, the bronchioles, and the muscles of the eye. The functions of smooth muscle are twofold: to produce motility, for example, to propel chyme along the GI tract, or to propel urine along the ureter, and to maintain tension, for example, smooth muscle in the walls of blood vessels. Smooth muscles are classified as multi-unit or unitary, sometimes called single unit, depending on whether the cells are electrically coupled. Unitary smooth muscle has gap junctions between cells, which allow for the fast spread of electrical activity throughout the organ, followed by a coordinated contraction. Multi-unit smooth muscle has little or no coupling between cells. A third type, a combination of unitary and multi-unit smooth muscle, is found in vascular smooth muscle. Unitary smooth muscle. Unitary or single unit smooth muscle is present in the GI tract, the bladder, the uterus, and the ureter. The smooth muscle in these organs contracts in a coordinated fashion because the cells are linked by gap junctions. Gap junctions are low resistance pathways for current flow, which permit electrical coupling between cells. For example, action potentials occur simultaneously in smooth muscle cells of the bladder, so that contraction and emptying of the entire organ can occur at once. Unitary smooth muscle is also characterized by spontaneous pacemaker activity, often called slow waves. The frequency of slow waves sets a characteristic pattern of action potentials within an organ, which then determines the frequency of contractions. Multi-unit smooth muscle. Multi-unit smooth muscle is present in the iris, in the ciliary muscles of the lens, and in the vas deferens. Each muscle fiber behaves as a separate motor unit, similar to skeletal muscle, and there is little or no coupling between cells. Multi-unit smooth muscle cells are densely innervated by postganglionic fibers of the parasympathetic and sympathetic nervous systems, and it is these innervations that regulate function. Excitation-Contraction Coupling in smooth muscle. The mechanism of excitation-contraction coupling in smooth muscle differs from that of skeletal muscle. Recall that in skeletal muscle, the binding of actin and myosin is permitted when calcium binds troponin C. In smooth muscle, however, there is no troponin. Rather, the interaction of actin and myosin is controlled by the binding of calcium to another protein, calmodulin. In turn, the calcium-calmodulin complex regulates another protein known as myosin-light-chain kinase, which regulates cross-bridge cycling. The steps involved in excitation-contraction coupling in smooth muscle are illustrated in Figure 1-28, also found on page 40 of your book, and occur as follows. Number 1. Action potentials can occur in the smooth muscle cell membrane. The depolarization of the action potential opens voltage-gated calcium channels in the sarcolemmal membrane. With the calcium channels open, calcium flows into the cell down its electrochemical gradient. This influx of calcium from the extracellular fluid causes an increase in intracellular cytosolic calcium concentration. Two additional mechanisms may contribute to the increase in intracellular cytosolic concentration: ligand-gated calcium channels and inositol 1,4,5-triphosphate or IP3 gated calcium release channels. Ligand-gated calcium channels in the sarcolemmal membrane may be opened by various hormones and neurotransmitters, permitting the entry of additional calcium from the ECF. IP3-gated calcium release channels in the membrane of the sarcoplasmic reticulum may be opened by hormones and neurotransmitters. Either of these mechanisms may augment the rise in intracellular calcium concentration caused by depolarization. These are again summarized on page 40 of your book in figure 1-28. Number 3. Rise in intracellular calcium concentration causes calcium to bind to calmodulin rather than troponin. Like troponin C in skeletal muscle, calmodulin binds 4 ions of calcium in a cooperative fashion. The calcium-calmodulin complex binds to and activates myosin-light-chain kinase. 4. When activated, myosin-light-chain kinase phosphorylates myosin-light-chain. When the myosin-light-chain is phosphorylated, the conformation of the myosin head is altered, greatly increasing its ATPase activity. In contrast, skeletal muscle myosin ATPase activity is always high. The increase in myosin ATPase activity allows myosin to bind actin, thus initiating cross-bridge cycling and production of tension. The amount of tension is proportional to intracellular calcium concentration. 5. Calcium calmodulin, in addition to the effects on myosin described above, also has effects on 2 thin filament proteins, calponin and caldesmon. At low levels of intracellular cytosolic calcium, calponin and caldesmon bind actin, inhibiting myosin ATPase and preventing the interaction of myosin and actin. When the intracellular calcium concentration increases, the calcium-calmodulin complex leads to a phosphorylation of calponin and caldesmon, releasing their inhibition of myosin ATPase and facilitating the formation of cross bridges between actin and myosin. 6. Relaxation of smooth muscle occurs when the intracellular calcium concentrations fall below the level needed to form calcium-calmodulin complexes. A fall in intracellular calcium concentration can occur by a variety of mechanisms, including hyperpolarization, which closes the voltage-gated calcium channels, direct inhibition of calcium channels by ligands such as cyclic AMP and cyclic GMP, inhibition of IP3 production, and decreased release of calcium from the sarcoplasmic reticulum, and increased calcium ATPase activity in the sarcoplasmic reticulum. Additionally, relaxation of smooth muscle can involve activation of myosin-light-chain phosphatase, which dephosphorylates myosin-light-chain, leading to inhibition of myosin ATPase. Mechanisms that increase intracellular or cytosolic calcium concentration in smooth muscle. During the action potential in smooth muscle, calcium enters a cell from the ECF via sarcolemmal voltage-gated calcium channels, which are opened by depolarization. As already noted, this is only one source of calcium for contraction. Calcium can also enter the cell through ligand-gated channels in the sarcolemmal membrane, or it can be released from the sarcoplasmic reticulum by an IP3-gated mechanism shown on page 41 of your book in figure 1-29. In contrast, recall that in skeletal muscle, the rise in intracellular cytosolic calcium concentration is caused exclusively by depolarization-induced release from the sarcoplasmic reticulum. Calcium does not enter the cell from the ECF. The 3 mechanisms involved in calcium entry in smooth muscle are described as follows. Voltage-gated calcium channels are sarcolemmal calcium channels that open when the cell membrane potential depolarizes. Thus, action potentials in smooth muscle cell membranes cause voltage-gated calcium channels to open, allowing calcium to flow into the cell down its electrochemical potential gradient, as shown on the top right of figure 1-29 on page 41. Ligand-gated calcium channels are also present in the sarcolemmal membrane. They are not regulated by changes in membrane potential, but by receptor-mediated events, and this is shown on the bottom right of figure 1-29. Various hormones and neurotransmitters interact with specific receptors in the sarcolemmal membrane, which are coupled via GTP-binding proteins or G proteins to calcium channels. When the channel is open, calcium flows into the cell down its electrochemical gradient. And see chapters 2 and 9 for further discussion of G proteins. IP3-gated sarcoplasmic reticulum calcium channels are also opened by hormones and neurotransmitters. The process begins at the cell membrane, but the source of the calcium is the sarcoplasmic reticulum rather than the ECF. And this interaction between IP3 and an intracellular sarcoplasmic reticulum calcium channel is shown on the bottom left of figure 1-29. Hormones or neurotransmitters may interact with specific receptors on the sarcolemmal membrane, for example, norepinephrine with alpha-1 receptors. These receptors are coupled via a G protein to phospholipase C, or PLC. Phospholipase C catalyzes the hydrolysis of phosphatidylinositol 4,5-diphosphate, or PIP2, to IP3 and diacylglycerol, DAG. IP3 then diffuses to the sarcoplasmic reticulum, where it opens calcium release channels, similar to the mechanism of the ryanodine receptor in skeletal muscle. When these calcium channels are open, calcium flows from its storage site in the sarcoplasmic reticulum to the ICF cytosol. Calcium-independent changes in smooth muscle contractile force. In addition to the contractile mechanisms in smooth muscle that depend on changes in intracellular calcium concentration, the degree of contraction can be regulated by calcium-independent mechanisms. For example, in the presence of a constant level of intracellular calcium concentration, if there is an activation of myosin-light-chain kinase, more cross bridges will cycle and more tension will be produced. This is called calcium sensitization. Conversely, if there is activation of myosin-light-chain phosphatase, fewer cross bridges will cycle and less tension will be produced. This is called calcium desensitization. This concludes Chapter 1